Dr. Srinivas from G Healthcare, who is going to talk to us about DIGE technology and give us a demonstration on software to perform DIGE gel analysis.
The next one is Differential Gel Analysis, shortly we call as DIA. In this, you can see we can create a new project, new DIA here, like there is an option create workspace. From here you can, now it will take you to where we already saved our gels in our database. Now from the database we can select any particular project, and from there I am selecting gel number 1 as we saved this one. Then now you can see this gels where we have uploaded. So now after uploading here you can process these gels. Then during process you have to give some number. This is some threshold which you are giving here. Actually this may be 2000, so that it will takes care of background issues also. Then basically in Decider code detection will be happens, but I would like to explain you some more about what is code detection. This code detection is uses a information of all 3 channels and will create a geometrically identical spot boundary for a spot across all the channels. That means there are 3 channels, side 2, side 3, side 5. Out of these 3 channels, in side 2 image it creates a particular volume, and the same area can be applicable for the remaining 2 gels also. This is way it works. In this way quantitative and qualitative results are much more accurate than with a single detection. In DIA, each image is co-detected with its internal control, producing 2 images pairs. The ratio of standard sample is calculated further, or the ratio of standard sample is calculated for each protein in each image. So as we see here, these are all the number of spots which it has been detected, in which there are, you can see some red color spots here. These are all down regulated and compared with control with treated, or and these blue colored spots where you can see these are all up regulated spots when comparing with control and treated. In between this there is a blue, there are some blue colored spots. These are all similarly regulated. So this is what we can see in DIA. Now you can go through each and individual spot and you can see the 3D view of that particular spot. If suppose if you can select any one, you can go to one by one and you can see whether it is exactly spot or some background, or if it is a background you have to remove that. So if suppose this is the background, so there is no spot at all, still it is detected some background. So you can exclude it from here by clicking, then confirm it. So this protein has been removed from the gels. So the same way you can go each and individual, then exclude it, confirm it. By this way you can check all spots and you can have more accurate data with you. Like you can see the how accuracy would be there, like the spot detection. Now you can see this is what we will get in DIA. This DIA creation we finished. This is the BVA. BVA is nothing but biological variation analysis. One of the internal standard image is selected as a master image and all internal standard image matched into this. Samples standard spot ratio for each protein, each sample, then compared giving t-test value, fold changes, ANOVA values for each and individual protein. How to create BVA workspace? can open the BVA and create a BVA workspace and go to our DIA workspace where we have our DIAs. From there you can create, actually add it. One DIA I have added, and this is another DIA I am adding here. Minimum 2 DIAs we require for the BVA. So, we have 2 DIAs here and click on create. So, it creates a new BVA for you. Okay, this is the new BVA. From here, the first all Site2 gels are automatically goes to standard folder. There is a standard folder you can see, and remaining all gels remains in the unassigned folder, where we need to assign these gels as according to the gel type or sample type. Then you just have to click on add option. Now create a group, it may be control or treated. This first one is the control, and apply some color draft. Confirm it. Then another you can create like treated, then give some color draft. Confirm it. Now we have two folders, control and treated. So, as we have in assigned folder both control and treated, this control gels we can transfer into control folder by dragging those images, and treated gels we can transfer to treated folder by dragging them. Now we have our images here. After shifting the control to control and treated to treated, we have to match all gels. Just click on match and match all. Just match. The matching process has been finished. Now, as we discussed, in out of all standard gels, one gel selected as a master image. As you can see, the number 24 gel has been selected as a master, and it will compares remaining other gels with this master gel. So, now we have this comparison data. After that, we need to calculate statistical parameters. So, click on statistical parameter button. See, now we have some statistical parameters like independent t-test, average ratio, student t-test, one-way ANOVA in between different groups. We are doing in between control and treated. So, calculate them. So, the statistical parameters has been calculated. Now, we can see exact results of statistical parameters. If I can go to the table view, here you can see t-test, one-way ANOVA, this you can see. So, we can select from here which are all the statistical significant and which are all not significant. After analyzing the statistical data, now we can see the complete results here. Here we can see the 4 views, like this is the image view and this is the histogram view. In histogram view, we can see clearly particular protein how it is behaving throughout control and treated. If you can see, this is the standard gel, that means this is the mixture of these control and treated. So, it is somewhere 0 we can consider this one. Then the control is completely showing upregulation, and after giving the particular treatment, it is showing the down regulation. So, this kind of data we can see here. Then in the table view, as you can see the complete protein data, where the t-test value, average ratio value, 1D ANOVA value, this all we can see we can see here in the table view. So, the 4 views at a time to see, this is the 4 views we can see here. So, after this we can filter them according to our interest. So, select the few parameters which are available, restrict to 10 gels which are spot maps which are present, a particular protein should be there, and student t-test as well as average ratio, then one way ANOVA value, then filter it. So, there are 2299 spots are available in this all gels, but 107 proteins only passed all these parameters. So, these parameters we can select as a protein of interest and assign pick list, so that these proteins can be saved in a file. This file can be given furtherly to our spot picker. These are all the things we can able to identify very easily in BVA. This is very user friendly. There is no much more manual interference. Okay, this is what this helps you to analyze your DIGE gels.
So, can you elaborate on what is EDA or extended data analysis, what it can do, which we are unable to do it in BVA? So, there is the layers here, right? One is the DIA followed by BVA, and then ultimately EDA analysis.
Exactly. Basically, what we can do here is we can compare two BVAs together there. Exactly. Here we are talking about a particular disease or particular set of data only. There we can analyze different BVAs together in DDA. There you will get majorly differential expression, again you will get, as well as PCA and discriminant analysis. You will, these kind of statistical data you will get in EDA. Very shortly, just show you briefly regarding.
So if I understood correctly, probably the statistical parameter will be more stringent over there in the EDA, and you can have some better biological significance information from the data set.
Exactly.
Because in a lot of clinical data or different type of treatments, people like to do several gels and lot of treatment.
Yes.
So your number of samples to be analyzed is very large.
Exactly.
And really obtaining the meaningful information is one of the major challenges in all the proteomics field. So I'd like to see in an EDA.
Please. Thing which we can see here is differential expression analysis, in which you can see differences in between control as well as two experimental data. This is the different treatments which given. Here you can see how the particular protein is expressing throughout these control as well as this PF and PV, these kind of things, and you can see this kind of data for each and individual protein here, so that from here you can see which one is your interest and which is not
so
your interest.
You are actually analyzing the data spot wise now, spot by spot. You are looking at
exactly. Here the spot by spot which we are seeing, the number of the index number which shows there is a master gel. From that master gel you can see that exactly this number. This is what which we are seeing here for each individual spot. Here we can see the results. Then as well as if we can see principal component analysis for this data. Here there are almost 89 proteins. Out of these 89 proteins, you can see there are inner the circle there are proteins, some proteins are present, and outer the outliers are there. The inner circle they are similarly, especially if I can say 95% statistically significant there, and outliers which you can see are exactly this can be some non-reproducible spots, or else what the major thing is, these are very highly upregulated or highly downregulated. So this can be worked as a marker also. Then we have to go back to our BVA data and we can check the protein how exactly it is regulating. Then we can identify that protein and we can use for further analysis.
So this is a powerful statistical parameter.
Exactly.
By using which you can identify some outliers, which could be the potential discriminator between the control and treatment. And once you identify those proteins, then actually you can go back to your original data from the BVA, and get all the analysis done.
Exactly.
Oh, this is very interesting.
And the next pattern analysis, you can see the whole proteome, then how these are different from each other.
So this is the heat map of
this is
data.
the heat map of the total 82 proteins are which we take into consideration. Then how in which area they are upregulated. If you can see the blue area, the green area exactly we can see is completely downregulated area, and the red color portion where you can see that is the upregulated portion, and the remaining black color which you can see, so those proteins are similarly regulated. This is what which you can see here. This kind of data will help you to represent your complete whole analysis.
So Dr. Srinivas, it was very useful to have you here and to get an overview of DIGE technology, how people can use decided software and analyze their data by using DIA, BVA and EDA. And although there was not enough time, but you gave a very good demonstration in a very short time to give a glimpse of the processes involved for doing this analysis, as well as how different type of statistical parameters can be applied to get some very powerful statistical information from our biological data. So thank you very much for coming here and giving this very good introduction about DIGE technology. So I hope our discussion with Dr. Srinivas was useful, and now you can perform these analysis by using a specialized software and obtain some very useful biological information from your data set. Probably you must appreciate there are lot of meticulous steps are involved in performing these experiments, but at the end this provides a very useful quantitative multiplexing approach to separate proteins and to analyze different type of variations. I hope at the end of this module and lecture, you will be able to perform the gel based proteomics experiment. But please keep in mind, these protocols and methods are only giving you a feel for performing these experiments. Each experiment, each sample type, each biological question brings its own unique challenges, and depending upon those conditions and your sample type, you need to optimize these methods. There is no one technology which can answer all of your question, but it is good idea for you to know that what are different methods which are available for you to use. So, I hope by taking this module on gel based proteomics, now you are familiar with different type of gel based techniques. These are only few, there are many other methods as well available, but these are the most commonly used methods which people are applying in the fields of proteomics. So, among 2DE and DIGE, which of these two techniques will be better to separate serum protein samples obtained from large number of patients in a clinical trial? So, if you have multiple serum samples from patients, two-dimensional electrophoresis, although a very useful technique, but it may not be the best option in this case to analyze serum proteins from large number of patients. In this case, DIGE will be extremely valuable tool for analysis of large number of samples simultaneously without having to overcome the problem of gel to gel variations. In dye gels, the control and test samples can be differentially labeled by using the cyanine dyes and then run on the single gel. The pooled internal standard for DIGE is prepared by mixing equal amounts of all the samples that need to be run on the gel, and this prevents the problem of gel to gel variations. From the same gel, 3 different images can be obtained for psi 2, psi 3 and psi 5. Therefore, there will be no reproducibility issue and various artifacts can be eliminated for the clinical or large number of sample analysis. So, the main aim for the development of difference in gel electrophoresis was to overcome the inherently poor reproducibility of conventional 2-dimensional electrophoresis. So, DIGE is quite sensitive technique with less than 1 femtomolar of proteins which can be detected, and it can enable the linear detection of very broad dynamic range of the proteins. So, as you can see in this slide, the protein samples, let us say you have a control and a treatment, those are labeled with 2 different dyes, psi 3 and psi 5, but a small aliquot of both of these samples is mixed together to make a internal pool. That internal pool is labeled with third dye psi 2. Now, all these 3 protein samples are mixed together in one tube, which contains both control treatment as well as the reference spots from the internal pool. All these protein mixture are separated in the first dimension on the same strip, and then the same gel can be scanned with the 3 different wavelength to obtain the images for the psi 2, psi 3 and psi 5. So, in the conventional 2-dimensional electrophoresis, the gel to gel variations which comes from the acrylamide polymerization, electrical, pH and thermal fluctuations in different gels, that can be overcome in the dye gels, because all the protein separation is going to happen on the same gel. So, all those artifacts can be minimized by using DIGE approach. So, in the slide it is shown that the 3 samples are mixed and then isoelectric focusing is performed from the pooled sample, and then this strip is placed on the SDS page gel for the protein separation in the second dimension. So, overall DIGE provides very uniform staining from gel to gel and shows high sensitivity and linear dynamic range of detection for the expression profiling of complex biological samples. So, if your aim is to resolve thousands of proteins and cover comprehensive proteome coverage, then DIGE is a very good platform, especially if you want to do the comparative or differential proteomic analysis, because your gel to gel variations and other variations will be minimized and DIGE will provide the very high sensitivity. So, once you have run these gels, now from the same gel the images can be obtained, 3 images of your control and the treatment, and these can be analyzed from different software such as D-sided software, and then by looking at 3-dimensional views and the statistical data, then these proteins can be considered as interesting for further investigations. Once the spots are analyzed and excised from the gels, then the same tradition you have to follow. You can use any of the mass spectrometry platform and then obtain the MS spectra for further analysis using different type of bioinformatic tools such as Mascot. So, overall the DIGE method is far more superior in terms of the reproducibility as compared to the conventional two-dimensional electrophoresis and for the quantitative accuracy. So, therefore, applications of 2D-DIGE can be found in virtually all major biological research areas. If you see the recent publications, you will appreciate there are several papers on each of the biological system for different type of applications, whether it is cell signaling, it is looking at developmental biology, looking at plant proteomic analysis, neurosciences, clinical studies, different type of diseases including cancer. You will find there are hundreds of publications available which have employed the power of 2D-DIGE technique. So, let us talk now a new case study 3 on 2D-DIGE as a strategy to identify serum markers for the progression of prostate cancer. A study by Barney et al. Published in 2009. So, in this study authors aimed for identification of serum markers by depicting the progression of prostate cancer by using difference in electrophoresis technique. So, prostate cancer is recognized as a significant problem in older male population. The prostate cancer screening rely heavily upon testing for the higher level of prostate specific antigen, also known as PSA, within the peripheral circulations. So, PSA is very sensitive marker, but there are lot of discussion on reliability and the specificity of PSA for the prostate cancer. Reason being that the level of PSA is also high in benign prostatic hyperplasia or prostatitis. So, therefore, there is a lot of discussion whether one should rely on only PSA for detection of the prostate cancer. So, this study aimed to identify some new markers in the prostate cancer by studying the serum proteome analysis. So, as you are aware, and in fact, we have discussed the protein preparation from the serum earlier. So, each of the biological sample poses lot of technical challenges, and serum is one among them, where presence of highly abundant proteins such as albumin and immunoglobulin, they result into the masking of low abundant proteins. So, to eliminate those high abundant proteins, authors used multiple affinity removal system from the Agilent technologies, and they removed most highly abundant proteins from the serum sample, including albumin, IgG, antitrypsin, IgA, transferrin and haptoglobins. So, after the abundant proteins were depleted from the serum sample, then authors moved for the protein extraction and further analysis. So, the differential proteomic analysis was performed in the two different cohorts of histologically confirmed prostate cancer with different grades of the disease. So, they used the patients with two different grading system based on the Gleason grading. So, the Gleason grading system that is used to help and evaluate the prognosis of men with the prostate cancer. So, depleted serum samples obtained from the patients with Gleason score 5 and Gleason score 7 were used for comparison and further analysis. As you can see in the slide, these samples were first labeled with the Psi 3, Psi 5 and also the internal reference pools were made which were labeled with the Psi 2 dyes. These samples were then further mixed. The depleted cancer serum from first cohort of Gleason score 5 and second cohort of Gleason score 7, those were mixed, separated in the first dimension and followed by proteins were separated in the second dimension. Now, when authors analyzed these DIGE images, they found that 63 protein spots were differentially expressed between the Gleason score 5 and 7 cohorts, and 13 of these proteins were statistically significant among these two populations. So, as you know, analysis of these gels is always challenging, especially if you are looking at the conventional 2D gels, where you have separate gels obtained from each of these groups. But analysis in the DIGE gel is more automated. So, if you remember our previous discussion in the DIGE gel analysis, this analysis is more automated and more straightforward, but still you have to go through individual spots and you have to look for the how real, how significant those changes are, and you have to look at the 3D views of those spots to ensure that it is the reproducible among various control and treatment groups. So, the different level of analysis is performed, which we have talked earlier, but this just shows you the final output, that 63 spots after all the analysis steps were considered significant. After 2D-DIGE gel image analysis, authors excised those spots and subjected for the mass spectrometry identification of proteins. So, the proteins excised from the gels were analyzed by using Finnegan LTQ mass spectrometer, and data from these MS experiments were analyzed by using BioWorks browser by using Sequest program. For validation, authors employed various techniques including Western blots, enzyme linked immunosorbent assays or ELISAs, and also immunohistochemistry. So, the pigment epithelium derived factor PEDF and zinc alpha 2 glycoprotein, also known as ZAG, those proteins were further validated by the ELISA technique. So, the PEDF levels were quantified by using ELISA kit, and results demonstrated, as you can see in the slide, that the statistically significant decrease in the PEDF in the Gleason score 7 depleted serum group, whereas the results for zinc alpha 2 glycoprotein ELISA analysis, which is shown in the red in the bottom panel, that indicated a 1.4-fold increase in the zinc alpha 2 glycoprotein absorbance in the Gleason score 7 group. So, these studies, this ELISA validation confirmed their findings from the 2D-DIGE experiments. Authors also employed immunohistochemistry or IHCs for validating the pigment epithelium derived factor PEDF and zinc alpha 2 glycoprotein. So, that they are very confident that the proteins which they have identified from the proteomic profiling, those are real, and they also tested those on the independent tissue samples. So, from this paper, the major conclusions were that serum markers which are reflective of the pathological grade and stage could be beneficial for the identification of appropriate treatment strategies. Authors confirmed that differential expression of PEDF and ZAG can be performed by using various techniques such as Western blot, ELISA and immunohistochemistry. Based on their studies and the follow-up experiment, they concluded that PEDF could be a potential marker of early stage prostate cancer prediction. However, more studies and follow-up required on the large number of patients before it can be established as a good biomarker. You may appreciate that there is lot of power of these techniques, and these can be employed for any biological application. You pick an application of your choice, and I am sure you will be able to answer those by employing 2D or 2D-DIGE techniques. Thank you.